It's not connecting. No, it's actually acting as a point where genetic hormone and where your this is are actually attached by okay. Let's do it. Okay, so exam 2, people have been asking me if I'm going to change it. I think I might change it by 1 day, if anything. Well, as we get closer, so think of it as being April 1st. If for some reason we need the time, all right, we should do it April 1st. Maybe we should. Maybe I'll give you a surprise. If anything, I'll change it to the 3rd. What? What?
Hemimethylation.
Yeah. We're getting there.
Okay.
So yes. It was the 3rd, and I'm going to change it to the 1st. But if I feel concerned, I'll push it off to the 3rd. Huh? I can't hear you. The thing is, what might happen if we do it on the 3rd is that we'll lose time having nothing to talk about, and then later we'll have to go fast, which I don't like to do. I'd rather be slow and have us understand things and have to rush. So this is one of those things where people get frustrated because everything's not set in stone, and I'm sorry about that, but that's what happens when you reorganize a class. Did I see a hand up? No, but I feel like this is plenty of notice. You've got a month's notice. Okay, so just keep it in your mind, the 1st, but it might become the 3rd if it seems like we're too slow. Quiz error, right. So a very smart student who may or may not be here yet emailed me. Only one person emailed me, which concerned me. In bacteria, only fully methylated origins are available for replication. I had checked the wrong one. And so if you check that, you got it wrong. So I put in a regrade, so it should all be fixed by now. But just so you know, if you were confused, that's what was going on. Any questions about that? We'll talk about it in a minute. This is the correct one. Yes.
So people who put the other one and now get it wrong.
Yeah, because it was wrong.
I mean, I did.
Yeah. People who put the other one get it wrong because it was wrong. It was never correct.
People who put hemimethylated, right? They got it wrong.
People who put hemimethylated get it wrong. People who put fully methylated who got it wrong now will have it right. Yeah. I checked the wrong box. Okay. So far, this is our study guide. So at this point, we've gone through quite a bit of ESPAs after introducing a whole bunch of concepts involved in cell cycle regulation. And we've been talking about the proteins that are important for DNA replication to take place. So as we go through these, I'm just kind of adding them so you'll have kind of a list. Most of these are things you learned about before or things that you think about for 2 minutes make sense, so they shouldn't be too long. And they shouldn't, I hope, they shouldn't come down to memorization because, I don't know, a lot of them make sense. So let me know if you have a hard time putting them all together in some kind of functional model, and maybe I can help you. So last time we did helicase, single-strand binding proteins, topoisomerases, then we were starting to talk about the sliding clamp and clamp loader. And so if you remember, I was talking about how I have this really great demo, not really great, but like this demonstration that I wanted to do, and I'd forgotten my yarn in my office. So we're going to just quickly do it. This was when we were talking about topoisomerase. And I think in the past I've tried to have everyone have their own piece of string, but that like took so long to get that I was like forgetting. And so this piece of string has, well, it's actually 2 pieces wound around each other, right? So we're going to call it a double helix. And as you can see there, we're going to have Venus help. hold on to one end. And so, and then feel free to come up at the end of class. But like, so you can imagine if I'm the helicase and I am opening this up, you see what's happening? All this is starting to, hold it up in there. It's all like wound up around itself. This is this kind of torsional stress that you get from that opening. See that? And so the topoisomerase actually comes in and will either clip one strand, and then all that force that's kind of built up, it'll unwind, or sometimes we'll clip both strands, but that's in a situation where you have multiple strands, thank you, of DNA. So feel free to come up after class to experiment with this, but it's fun to play with yarn. And so now it's all a big mess. And so I need the topoisomerase to come clean it up. But I've got plenty of strength, so we can end a little early and you can come up and do it yourself. Yes?
So
Topoisomerase. Depends on which topoisomerase you're talking about. We learned about two. One, the type 1, I believe, is where it binds and it makes one cut, and then it rotates around the backbone that's still attached. And then the other type that we learned about is that strand passage, and that's where it makes the double strand cut. And that's in a situation where when you're done, you've got them all jumbled up together and all, and then it might be easier, it will be easier to kind of cut one and pass one through. So it'll bind to one, and it'll bind to one strand, and then cut the other strand, and then pass that first one through. Make sense? Yes?
So while like the DNA, are both topoisomerases, are they like could they both be used for like different parts of DNA? Like say
Yeah, so one, it doesn't mean like you're either going to use this or this. It's just depends on what's needed. So you can imagine cell. So the question is, tell me if I was jumping the gun. The question is, there are different types of topoisomerases you're talking about. Can both be used at the same time in different parts of the DNA to make things right. And really, just imagine the nucleus is a big bag of DNA and proteins, right? And it's full of all, like right before S phase, you can imagine gene expression of all the genes needed for S phase is going to increase. So you're going to have high levels of expression of topoisomerase, both kinds, and helicase, and all these different proteins needed for S phase. And so if that torsional stress is sensed, and don't ask me how it's sensed, then whichever one is needed. There's probably certain, we can probably look, actually, it's been a while. Is that a lot to ask for? Let me think for a minute. But as I was going to say, is there's probably different ways the cell has to detect different types of stress and different types of requirements for different topoisomerases, and the correct one will be recruited and act in the correct location. You can imagine that in eukaryotic cells with all these different chromosomes and all these different origins, there's a lot going on at once, and it's a mess. So you need all of these happening together. And that's pretty much how all of cell biology is we teach you about one thing and one thing and one thing, but in reality, it's all happening at the same time, right? The signaling molecules binding to the receptor at the same time that, DNA replication is starting. It's all happening at the same time. I was just thinking about whether I would make an extra credit, but it might be, because she wasn't truly stumping me, but it's been a while. And because I was thinking about how, even if I wanted to tell you how different topoisomerases were recruited to different sites where they're needed to put in, because they never studied that. And I'm wondering if it would be useful or interesting to have you guys do that. Because you guys just want extra credit. Okay, so it's not a true stump the teacher, because I basically stumped myself by deciding I wanted to understand something I didn't. And so I'm going to actually send you out into the world to do some research to find out how different topoisomerases are recruited to the sites where they need to be used, or need to be active. Does that make sense? The mechanism behind recruitment of topoisomerase to regions of DNA requiring topoisomerase activity. How's that sound? Everyone? Mechanism behind recruitment of topoisomerase to regions of DNA required regions of DNA that require topoisomerase activity. Remember, there's two types of topoisomerase, so there's some specificity here. There's some regulation because you don't want topoisomerase just chopping up and moving things all over the place. So there's, I'm sure there's a whole field of research that you guys can dive into. So whoever gets that to me by tomorrow night-ish, or night, if it's not, do not copy and paste. I keep forgetting to tell people, don't just copy and paste and send it to me. You actually need to write a coherent, and it doesn't need to be a Word doc, you can write right in the email. A coherent, and actually what I should do is set up a thing on Blackboard for you to do this, but I'm not that organized yet. And referenced. So I need references, and it can't be from a website about like longevity or something weird like that, because people do that too. So, good references, no copy and pasting, actually cohesive, I thought about this is the mechanism I found, here's the reference where I found it. For night, 1 point. Okay. Yeah. Okay. Let's start learning something. 10 minutes. Okay. So the high processivity, so we talked about processivity when we talked about DNA polymerases and how some polymerases are highly processive, meaning they stay on for a long time, they don't fall off. And some are distributed, so not highly processive, and they just go to where they need to do their thing, they do their thing, and then they fall off after a short period of time. Where are your highly processive polymerases? What process are they involved in? Right, DNA replication, replication of your whole genome. So it needs to go a long ways, right? Between origins. Yes. And what are your distributive polymerases involved in? Repair, mostly, yeah. Good ways to think about it. So this processivity is due to the sliding clamp. And so you can imagine the sliding clamp is going to associate with your replicative DNA polymerases. And so this is just showing the structure of the bacterial and eukaryotic sliding clamp. And you can see, would you say that the sliding clamp is fairly conserved? Do you guys know what it means when people talk about conservation amongst proteins? We're not talking about species and stuff like that because I don't understand that.
Same sequences among the, yeah.
Same sequences among different species, yeah. The shape stays the same. So sometimes the sequence might be different, but the structure of the protein is the same. Yeah. Anyone else want to add to that?
Would it mean
So it's used across different species, therefore that structure is very important. Yep.
Maintains the function.
It maintains the function in different species. You might guess that probably taking a highly conserved protein out of one species and putting it in another, it might work the same. Not all the time, but sometimes. And so this is interesting because it's showing, the way they show it here is just in the different, so they're made of polymers of different proteins. And so bacterial sliding clamp only has two types of proteins that are trimers. And then the eukaryotic sliding clamp has three types of proteins that function as dimers and all assemble together into a 2, 4, 6 hexamer. And so you can say right here, it's not the same because this has 3 different types of proteins, this has 2, but the structure is quite similar, right? And so this keeps polymerase tethered to the DNA. So that's what it looks like. These are right out of, this one's right out of the textbook. The clamp is the This is eukaryotic or bacterial? What do you guys think? Bacterial, right? Because it's showing two different types of proteins, and then the DNA is through it. So that's what the clamp looks like around the DNA. Okay, so the clamp is put on using the clamp loader, so that's another protein. And it binds specifically to the primer template junction. Does that make sense? So the clamp loader loads the clamp onto the DNA and the clamp loader specifically interacts with the primer template junction. I want you to stop and think about that. Does that make sense to you? What does that mean to you? Primer template junction. What am I referring to? Because this was, I didn't know this before I started teaching this.
Primase puts a primer.
Okay, primase puts a primer. So what is the primase primer template junction? So it's where the primer is bound to the template? Do people agree with me? So does it make sense that the clamp loader would specifically want to bind to this region? Why?
That's where you want to start
That's where you want to start
replication.
replication? So, okay, so you want the polymerase to be added at the primer template junction? Yep.
Does it have anything to do with the 3' end?
This is actually showing how, in this specific picture, this is showing the clamp loader, and there's the clamp, and there's the DNA. It's showing that the structure, and this is not really much of a structure, the model of the structure looks kind of like, I think they refer to it like a nut or like a washer or something like that. But basically, what it does is it fits in the grooves of the DNA, and you can imagine like a screw, and how you would twist a nut around a screw, and it would stay in the grooves of the screw. And so it's trying to show that it interacts kind of with the DNA that way. But specifically, it has to do with this primer template junction. Remember, I'm just going to zoom in on this for a minute, because this was one of these moments. I didn't know this, and then when I was reading and preparing these lectures, I read this and I was like, oh my God, that makes so much sense, right? Because there's your replication bubble. Oops. And so let's say this is a template primer. This is your primer template junction, right? Right? Do you guys believe that? And here's your 3' OH. Okay, so it likes to bind, so you're saying your clamp loader likes to bind over here and load your clamp right here and recruit your polymerase here. Does that make sense? It makes a huge amount of sense, considering that the first base is going to be added to that 3' OH. Right? If you just threw it on anywhere, if you just put it on over here, what's it like? Okay, yep. Yeah. You mean you don't like my donut approach? Here's your CL for clamp loader, and it recruits your clamp. And we'll talk about exactly how in a second. Loader, and there's your polymerase. I mean, I'm just drawing like a artist rendering, but like when I read this, it suddenly made so much sense to how it all kind of comes together. So it's nice to stop for a moment and reflect on this sometimes. Are you taking a picture? Okay. Anyways, we'll move on.
Why we only have one clamp loader because it's on the lagging strand?
Is that why we only have one clamp loader? Because so it's on the lagging strand. Well, let's talk about the clamp loader a little bit more. And I want you to think about that, and then we'll come back to your question as soon as we get through the next few slides.
Isn't that on this picture, the proportions are kind of out of range because they want to emphasize that?
Well, here, yeah, this picture is only showing how your clamp loader and clamp are interacting with the DNA. But let's move forward, because this picture is showing a little bit more about how it works. So you have you've got your sliding clamp and your clamp loader, and it uses ATP. So the ATP binding is required by the clamp loader, is required to bind to the clamp, and then it puts it onto the DNA, and then the ATP is hydrolyzed, which allows the clamp loader to leave. And then once your clamp is there, then DNA polymerase is recruited. And so you can kind of start to fit that into the model of what we've drawn in recitation, right? So every single time your DNA polymerase needs to come off, your clamp is going to be removed, your DNA polymerase is going to come off, and then your clamp loader is going to come bind to the next primer template junction, bring the clamp, recruit DNA polymerase, and then it's going to go until it runs into the next primer. So when you start to think about the lagging strand, and you're having, in reality, yeah, it's one clamp, because it's one clamp per polymerase. And then the clamp loader is really just loading and leaving. Okay, does that address your question at all? Okay. So the clamp loader is released once the clamp is assembled, but it stays nearby the lagging strand polymerase because it's ready to assemble a new clamp at the start of each Okazaki fragment. Yes?
So the clamp loader isn't essentially a part of the whole structure.
The clamp loader is hanging out there with the helicase. And we'll see a picture and I'll show you where it is. And so it's hanging out there, and every time it's time to place the polymerase on a new primer in the lagging strand, it comes and it does that. Yeah. Any other questions? Okay. So this is just another picture from, sometimes I'll put multiple pictures from multiple textbooks in because some look nicer than others. So here it's showing clamp loader. So you've got your clamp loader, and then the ATP binds, and that leads to a conformational change that allows the clamp loader clamp to interact and open. So remember, we have to open the clamp. And then you've got the 3' end of your primer, and then you've got ATP hydrolysis. So there's your primer template junction there. Then you've got ATP hydrolysis, which makes the whole thing go away. And then DNA, the clamp itself recruits DNA polymerase. It all makes a lot of sense when you think about it. Okay, so let's talk about the mechanism. We've talked a little bit about this, but we'll just talk about initiation here for a minute. Well, so to go back, like, you can break your replication mechanism up into these major parts, right? Initiation, priming, leading lagging strand synthesis, expansion of your replication fork, and then termination, which is essentially when you run into your other replication forks. Okay, so this is kind of the basic picture. The same kind of thing happens eukaryotic and prokaryotic to a point, but the players and the regulation differ. Right, so you have your origin, and your origin is going to contain some site that binds to origin proteins, the ORC, origin recognition complex, or some initiator proteins, and then some AT-rich stretch of DNA. Why AT-rich? Yeah, easy to open, double bond versus triple bond in GC. Yeah, exactly. So AT-rich, easier to open. And so this is all loaded before DNA replication is ready to begin. And so then the complex is activated somehow. In eukaryotic cells, how did we learn this is activated? By what? By what kinase? by SCDK, right? So this is, so if you're going through all this and thinking to yourself, how am I going to bring this all together? All that stuff we talked about, and we're going to bring it back today also, about cyclin, CDKs, all that stuff. Well, this CDK is important in phosphorylating things and activating things. And so when this complex in eukaryotic cells is activated by SCDK. So this is showing your initiator protein binding, and then you get separation of your AT-rich strands, and then loading of your helicase, and then you get your other replication proteins. This is very general. Yes? Yeah, so remember we talked about this, this is specific to eukaryotic cells, what I'm going to say right now. Remember we talked about this pre-replicative complex? Right. So all that is set up and waiting at the origins until SCDK is active. And then SCDK, once it's active through cyclin binding and the proper phosphorylations, dephosphorylations and all that, then once SCDK is active, it can phosphorylate things, which will change the conformation of that pre-replicative complex and recruit polymerase, and actually the origin will fire. Okay? And so we'll go through that a little more right now. But so this is where you should start bringing all that CDK stuff together, hopefully. The origin recognition complex is recruited to origins with hyperacetylated histone tails. Also, this is specific to eukaryotic cells, right? Why hyperacetylated histone tails? Well, would it make sense to try to have an origin with hypermethylated histone tails? Would you ever be able to open up anything? No. So that's pretty logical. Okay. I'm going to take a break. If you haven't noticed, I'm sick. I was reminding someone, I guess it was Ashley, a student I had previously who was in my class. It was the end of the semester, and I was so sick. I always lose my voice. It's the first thing that happens. And I lost my voice so badly that I was like, and everyone had to like sit in the first 2 rows because I could barely talk. But I didn't want to cancel class because it was like the second to last day before the final. So we won't let that happen. No, never again.
Yeah, spring break.
Spring break, I can recover. Okay, so DNA polymerase functions as part of a large multi-protein complex. You know that, you drew it, you understand it, you love it, right? This is what we're talking about. So this is where all that comes into play. I'm not going to go over it because we've spent plenty of time on it, right? Unless there's any remaining questions. Yeah. Okay. So in bacteria, it's looking quite similar. There's a simple origin, again, with the initiator binding site and this AT-rich stretch of DNA and some kind of initiator protein that's going to bind and participate in opening up the origin. Yes?
So how does the attractor attract the initiator protein?
How does the initiator attract the initiator protein? Is that what you're saying?
No, I'm confused about the attract initiator.
Oh, so you're wondering how initiator proteins are attracted. Okay, so we're going to talk in a second. I don't go very deeply into bacteria because, we're going to talk about methylation status, and that's how that partially regulates this process. But just again, like what we said before, right, AT base pairs are easier to open than GC, right? So there's your AT bond, only 2 bonds, whereas GC has 3 H bonds, so it takes more energy to open. Okay, so this is again just a basic process of replication fork initiation, which is also fundamentally the same in bacteria and eukaryotes. right, where you'll have the opening. So of course you would imagine you've got your initiator proteins bound, your AT-rich region, that's all the same. Then you'll get primase will come and you'll get primer synthesis, and then you'll get leading and lagging strand synthesis beginning, and then your two replication forks are going to go in opposite directions. You'll get leading and lagging strand synthesis. This is what you've learned like many times already. So this replication fork initiation forms a replication bubble. So often people will be like, okay, what's the replication fork? What's the replication bubble? So with replication fork, it's specifically this thing right here. You can think it looks like a fork. I guess it depends on which way you look at it. Maybe it's like a two-pronged fork, right? Right here. That's your fork. One fork, two fork, right? And replication bubble is the whole thing. It looks like a bubble. For some reason, I get that question a lot, so I just want to clarify it. Okay, so in bacteria, you only have one origin. And again, we learned that again in recitation, right? When we were reading the paper about the timing and location of origin firing. And I asked you guys whether that would be a useful study to do in bacteria. And most of you said no, because there's one origin. So only one replication origin in bacteria, which means there's two replication forks, one replication bubble, and then there's two DNA polymerase enzymes at each replication fork, right? Which means that there's 4 DNA polymerase enzymes per genome per replication cycle. Does that make sense to everyone? Yes? Yep. If I could draw it in.
Okay.
Yep, one would be here, one would be there, one would be there, one would be there. Leading and lagging. So remember, you have leading and lagging for each fork. Yep, so what I taught you guys when I had you draw, I had you focus on one fork. But you need to always remember that there's 2. So with, yeah?
This might be a problem, but like when all 4 DNA polymerase, don't they like eventually crash into each other and then?
You mean when they come around the other side?
No, because I mean,
They have many bubbles and they're open, like how
Are you wait 1 second. Are you talking about, excuse me, when, are you talking about eukaryotic or prokaryotic? Or just in general?
I mean, both have 4. I'm assuming they just.
So, like, both have 4 at each bubble, but they're going that way.
Oh, they're not. Oh, okay, that's why
Yeah.
Do you still get the trombone in this?
I guess so. Is this, yeah, why not? So do you need is the trombone model applicable to E. coli? Well, there's a leading strand, there's a lagging strand. The whole complex moves in the same direction, so why not?
And
Right.
it still has the same like complex, right?
Yeah, but I think the general picture that we drew, I believe applies to both. Yeah. Any other questions? Okay, so initiation with E. coli, a little bit more specific, right? So you have this AT-rich region and this initiator binding protein. And so there's a protein called DNA A, which is this initiator protein. And it's going to bind to that initiator or DNA A binding region, and it kind of distorts the DNA there. And what that does is that bending actually weakens the interactions at that AT-rich region, and so that allows more easily to open up the origin. Yes? So you can see these green balls. So those are DNA A, which are these initiator proteins, and they're going to bind to the DNA. And you see how it's all twisted up like that? They kind of act to distort the DNA. And so you can imagine that the distortion of the DNA over here will affect the DNA over here.
That's how it breaks it into AT.
And so that's how that encourages opening of that AT-rich region. Okay.
So
Uh,
do they have, I'm sorry.
Yeah, go
Do they
ahead.
have topoisomerases?
Do they have topoisomerase?
Is this acting in place?
So we're not there yet. They have DNA gyrase. And at this point, you don't need it yet because you're still just opening. But yes, they do have to deal with all that torsional stress because they have a double helix, right? And they're still going to have that opening and that, yeah. So they're going to need that kind of activity. Yeah. And not to forget that when you're done replicating your DNA, if you're E. coli, your two genomes are intertwined. And so we'll need topoisomerase to resolve that. And I'll show you that when we talk, hopefully today, maybe Thursday. Yes?
Right. So the E. coli is in eukaryotic, is it? Sorry.
Huh?
Is
E. coli is prokaryotic, is bacterial.
Oh, so would initiator proteins that those wouldn't be in eukaryotic, right?
There are initiator proteins in both. They're called different things, but they serve a similar purpose. Similar. Which really it's about opening up the DNA loading helicase. Big picture, it's all about opening up the origin to load helicase. So you can get as detailed as you want in it, but if you keep that big picture, it's like we need to open the DNA to load our helicase, and if you remember, helicase opens up the DNA to encourage replication, then you're good, right? Okay, so then you've got this open, and then DNA-C in this case is your helicase loader. Helicase loader in eukaryotic cells called something else, MTO complex. Don't worry about it. Helicase loader, right? It binds to and loads helicase B, which is the helicase. So it's just about getting that helicase loaded onto the DNA. And then DNA-C, which is the helicase loader, dissociates. And then primase and other proteins are recruited to make a primer, initiate DNA replication. Yeah. Oligomerize. No, oligomerize means multiple proteins binding together. It's kind of like dimerize means two proteins binding together. Trimerize means three proteins binding together, and so on. But oligomerize means many proteins binding together. Yeah.
So, just one more time, I'm sorry. Are you saying that DNA B is the helicase and DNA C is the helicase loader?
DNA-C is helicase loader, and DNA-B is helicase. But basically, try not to get bogged down with what does what. I give you this information, but really, if you can understand the helicase loader is going to load the helicase, then you're getting the big picture, you're understanding the basic concept. Any other questions? I feel like I saw another hand up somewhere. Okay, we're happy. Okay. Okay, again, this is just another way to look at it. This is out of our textbook. Again, you've got initiator. In our textbook, they talk about DNAG, which is primase also. But really, it's the same thing where you've got your replication origin with your AT-rich sequence. And then you have these initiator proteins that distort the DNA, allowing the helicase loading protein to kind of come and load your helicase. Here they're actually showing it in a slightly different order, where they're showing that helicase loading protein and helicase have something to do with opening up DNA. I guess we have to figure out which is more accurate. It might have to do with which book is newer. But in reality, it's about distorting the DNA so that AT-rich region can open and helicase can be loaded. Then primase can come in. So remember we talked about how primase is interacting with helicase. And so in this picture here, it's showing primase binding right there to helicase. And there it can lie down that first primer. And then you're going to get recruitment of your clamp loader, brings the clamp, and then your polymerase comes, and then you get replication in opposite directions. So this happens for all 4 chains of DNA that are being made. Okay, so we're just looking kind of in one spot here, but in all places you're going to have, well, this opening up is one thing, but the primase, the helicase, the polymerase, the clamp, that's all going to be recruited specifically. You know what? That's not logical. It's one. No, that says it in the book, but I don't believe that's correct. You will have different helicase, not polymerase for each and clamp for each, but the other ones, no, it's per form. I'm gonna have to write a letter. It's funny because I read this in the book and I was like, that doesn't sound right. It doesn't sound right. Anyway, go ahead. This is E. coli. This is bacteria. I'm just showing you a different way of looking at it. So sometimes I'll show 2 different figures to illustrate the same thing because one might speak to one student more than another. Yep. So here I wrote E. coli, here I wrote bacteria, but yeah. Helicase, one note is that helicase loading protein inhibits helicase activity until it's loaded on DNA. That's useful, right? Because you don't want helicases just going around and opening up DNA all over the place. Okay, so I was being asked before about regulation of initiation of bacteria. And so this is regulated very much by methylation status. So it's regulated by the availability of the origin for interaction with initiator proteins. It's as simple as that. It's not nearly as complex as in eukaryotic cells where you've got all of these things that need to be set up and all these proteins that need to be phosphorylated and degraded, and then other proteins need to be recruited and phosphorylated and activated. No. Here it's a little bit simpler because you'll have an origin, and if it's fully methylated, then that's the way for the cell to know, okay, we can replicate our DNA. And then after you get replication, you're going to end up with hemimethylated origins, right? Because there's a delay in methylation. Methylation doesn't just happen at the same time as DNA polymerization. And so you'll end up with your origin being methylated, but your newly replicated strand of DNA is not methylated. So that results in you being hemimethylated or half methylated. And those hemimethylated origins are resistant to initiation, so you cannot replicate your DNA if you have a hemimethylated origin. Did you get my email? Yeah, okay. My student who corrected me on this. Yes?
So, in bacteria, it's kind of the opposite of what we're learning in eukaryotes, where methylated means
Right, so there's a few things. Methylation, in terms of gene expression, is different than when you're talking about, so you can think about it this way. Methylation, to think of it very broadly, changes the shape of something, and it just so happens in bacteria, remember bacteria doesn't have histones or anything like that, and this is just DNA that's methylated, it just so happens that these initiator proteins like to interact with fully methylated origins because something about that structure binds nicely with those proteins. Whereas a hemimethylated origin is not going to bind, is not available for binding. And so over a period of time, the methylation will catch up, and then you'll have your fully methylated origin, and then you're ready again for replication to happen. And remember, bacteria, specifically E. coli, is it's dividing every 20 minutes. So it kind of makes sense that it's a very simplistic kind of system. And so regulation of methylation, you can imagine, it has something to do with the environmental conditions and nutrient availability. So you might think that methylation might be a bit slower when nutrient levels are lower. So you can think of this as a refractory period. It's a period where you have this delayed methylation and that prevents initiation of new rounds of replication. So does this answer the question you had earlier? if you remember it.
Yeah. Yeah.
About bacteria and how the regulation. Yes.
Can you like get rid of the factory period if you just increase concentration of DNA methyltransferase?
Can you get rid of the refractory period if you increase the concentration of DNA methyltransferase? Well, I guess it depends on which enzyme exactly methylates origins versus other types of methylation that happen for other reasons. But if you're talking about the DNA methyltransferase that specifically methylates origin, I would imagine that the whole way this is regulated is by regulating expression of that DNA methyltransferase, and how levels will probably be high when the activity should be high. Or it might be the enzyme might be regulated in a secondary way, but I'm not sure of that. But yeah, you're right. Overexpression of the enzyme would probably increase the enzymatic activity. Yeah. I think it's logical. Any other questions? Okay, so now let's go on to initiation of eukaryotic cells. And so this is similar to what we've spoken about a little bit earlier, I guess last week. So it'll look a little familiar, but I'm trying to tie it all in with initiation. Just a reminder, there are multiple origins in eukaryotic cells. We learned about that when we were talking about that paper in recitation about in yeast where they were studying all the different origins. And so this is a similar type of, this is one of the original, I guess, experiments that they did. And this is out of the textbook, this is not from the paper, where they were studying that, where they discovered that there were multiple origins in eukaryotic chromosomes. And what they did is they used tritiated thymidine, so radioactive thymidine, to label cells in culture. And then they lysed the cells and they stretched the DNA out on a slide. And then they were able to detect origins by autoradiography. And so you can imagine that your origins are going to start small and get bigger, right? And so if you start DNA replication and then you label for a certain period of time, here there's like 10 minutes, you're going to get labeling of that bit of DNA that's being replicated at that time. And then you'll get rid of your radioactive label, and then you'll get an expansion of that bubble. And you can actually watch over time the expansion of this bubble. This is even more familiar, hopefully, to you guys, where they studied using the heavy and the light media. And they followed replication over a period of time, each time taking their replicated DNA and hybridizing it to a microarray. And they were able to kind of follow which origins fired when. We did this in recitation. So these are just some fun facts about eukaryotic origins, right? So there's many per chromosome. Somewhere in the textbook it says something like eukaryotic chromosomes are so big that it would take, oh my god, it's some insanely long amount of time if there was only one origin. I have to look it up. I read it today. I meant to put it in the lecture because it was mind-blowing. Clearly not mind-blowing enough for me to remember how long. But it was a really long time, which is why you need multiple origins. It has to do with the size of the genome, right? So origins tend to be activated in clusters. And so you can think of them as replication units. So that would be multiple origins, 20 to 80 origins that would fire together. They tend to be activated at different times during the cell cycle or during S phase. Right? And then origins tend to be spaced within these replication units, anywhere from 30 kb to 250 kb apart. kb, kilobases. 2 replication forks per replication bubble, and then they move outwards until they merge with another bubble. Okay, so this again, big picture. similar to what you saw with bacteria, where you've got these AT-rich DNA elements and the initiator binding site, and all that's the same, but it's more specific, right? So, in terms of eukaryotic cells, this is very much regulated by not just methylation, like in bacteria, but it's regulated by different kinases, specifically SCDK. So here, your origin, your initiator protein is called ORC, origin recognition complex. It should hopefully look familiar because we've learned this already. And then here here are some of the other proteins that are involved, and then eventually loading the helicase. So again, it's about binding to your origin and then loading your helicase. Same concept, more proteins, more regulation. So you've got the CDC6, CDT1, and then MCM, and they're all loaded onto the DNA, so then you've got your helicase loaded. And then your MCM complex will become an active helicase only during S phase. So it's active only when SCDK is active. So there are different levels of regulation here. So I want you to start thinking in your mind, compare and contrast bacteria and eukaryotic cells. Because it's always nice to compare things. It's a nice way to learn. And so with bacteria, what's our main level of regulation? Methylation, right? And so here we're already starting to see different levels of regulation, right? You've got helicase loading and then helicase activation. That's two things, right? So the helicase can be loaded, but it won't be active. Yeah, it's not until it's activated that you have anything happening. So yep.
How would you fit into DNA, the other slide we talked about before, we showed DNA polymerase and loading time?
Okay, so you want to know about when the DNA polymerase and loading plan will come. Okay, so it's coming soon. So now we're just at, okay, so the question was, where does DNA polymerase and the clamp loader and the clamp come in? And we're not there yet. Right now we're just at that point, like similar to when we talked about a bacteria where we're binding to origin and opening up and loading helicase. So we're not there yet, but it's coming. But we do need to regulate this pre-RC assembly because this very much controls the frequency of DNA replication. And so S phase only needs to happen once per cell cycle, right? You don't replicate your DNA in S phase, then in M phase, then sometime in interphase. You don't do that. It only happens once per cell cycle. And a lot of that has to do with regulation of this pre-RC complex. So it's regulated at 3 different levels. And it mostly has to do with preventing assembly of this complex. So when I talk about the complex, I'm talking about assembly of this complex right here. So one is, so MCM2, that's the helicase. It is prevented from being loaded by a protein called geminin. And geminin is degraded by APC normally. So remember we talked about APC, ubiquitin ligase. So at the end of M phase, so remember, cell cycle is a circle. At the end of M phase, APC is active, and APC degrades geminin. And so now this allows this whole complex to set up and recruit helicase. But then later, APC is going to be turned off. So after S phase begins, APC is turned off, and then geminin levels are allowed to rise again. So then you get this inhibitor bound, and so you cannot load helicase again until M phase is over again. So that's one level of regulation. Another level of regulation is the CDT1 right? This protein here that's involved with loading helicase. CDT1 is ubiquitinated and degraded by SCF. And so that's due to phosphorylation by SCDK. And so when CDT1 is degraded again, you can't load helicase. Yes?
I'm gonna answer then because
Okay. So that's one. So all this has to do with this pre-RC, this complex of proteins that's job it is to load the helicase. And so then another level of regulation. So you had one level of regulation early in S phase that has to do with loading helicase. Another level of regulation that actually CDT1 is gone, so it can't set up again. And that's during S phase, because that's regulated by SCDK. And then another level of regulation with SCDK where it starts phosphorylating all these different components of this pre-RC complex so that they can't assemble at the origin. So it's 3 different levels of regulation that control whether or not you can prepare to begin S phase. And so that's why S phase, it starts, it goes through, and it doesn't start again until it's time, which is basically when SCDK, a cyclin is degraded, so SCDK is inactive, and APC is turned on, so geminin is degraded, basically. Okay. You had a question?
it was the origin of the ORC and then the CD, that whole complex, when geminin is a part of it, it's inhibited and when it's made, it's not inhibited.
Right, so geminin is going to inhibit specifically inhibit CDT1 from loading helicase.
Okay.
Yeah. Yeah. Any other questions? The whole entire thing?
Just you said geminin inhibits CDT1.
Geminin's job is to inhibit CDT1. So as long and CDT1 is involved with loading helicase. And so as long as geminin is there, helicase can't be loaded. If you can't load helicase, can you replicate your DNA? No. Yes.
So the geminin is present during M phase because you don't want DNA replication during M phase.
Yeah.
Not during S phase. So the geminin isn't present during S phase, but is during M phase.
So geminin is degraded by APC, and APC is turned on at the end of M phase. So basically, metaphase anaphase transition, APC is turned on, and that degrades geminin. And so now you can actually start to set up for S phase. Set up. That really just means assembling your pre-RC. Notice that happens in G1. But it's not activated until S cyclin rises to high enough levels to activate SC.
Okay, so it's not inhibiting the helicase during S phase, but it's present during S phase.
Geminin is degraded during S phase. That's why it's not inhibiting helicase. Make sense? Yeah. Okay. Any other questions? Yes.
Is this all part of one thing?
These are different levels of regulation of this pre-RC complex. So the pre-RC complex, its whole job is to load helicase. And so a lot of regulation of S phase actually comes with whether or not helicase can be loaded. That's one level of regulation. And then the other one will be whether it's activated. And so these are 3 different levels of regulation. And you can kind of see they're kind of at different times in the cell cycle.
So can you explain C then?
So C, this is happening actually during G2, S and then G2 really. And there are CDKs, S, I guess. Well, there are CDKs that are going to phosphorylate all the components of this complex. And so phosphorylation, remember, changes the shape of proteins. And so if normally they're unphosphorylated forms are what forms this complex, when they're phosphorylated, they're not going to form the complex. So that's going to prevent them from assembling until they're needed. which is pretty much going to be G1. So you can imagine there's going to be some dephosphorylation event that's going to happen later. We're not going to talk about that. Yeah. So this is how complex cell cycle regulation. This is not even close to how complex. But this is just like a sneak peek. Okay. Any other questions? Yes.
All of the primates changing the structure of helipase, so it binds to other than other side of that, not it doesn't bind to the active site of the other part of it. It changes the structure and stops the whole thing.
Okay, so let's talk for a minute. So helipase, primates. Go again. Come again.
Okay, so the helipase is actually part of the whole. The helipase and primase is like they bind together, but during the whole like DNA replication, primase stops, like changes structure of helipase. So what kind of regulation is that? Is that the same?
So that's different because here what we're doing, we're talking about regulation of initiation. That is actually during replication. So the question was about, you know, the interaction between primates and helipase and how that regulates DNA replication. And that what it's doing is kind of slowing, well, it's kind of slowing things down so your leading in ligand strand synthesis can be equal. But that, we're not even there yet here. This is really about setting up so you can load your helipase. Yeah. Any other questions? Okay. This is just another way to look at it. So phosphorylation by these CDKs also inhibit pre-RC formation. So I'm harping on the whole pre-RC formation because that is the main mechanism behind regulation of the frequency of S-phase in eukaryotic cells. So we'll just move on. Okay. So now we're all set up, right? Remember we were over here in G1. We have our helicase loaded at our origin, but it's not active. So then our S-cyclins rise. They bind to our SCDKs. Our SCDKs become activated, and they go and phosphorylate things. And so SCDK is what triggers S-phase and actually establishes active replication ports. And so you get phosphorylation of multiple things, including CDC6 and ORC, and then you get recruitment of polymerase, primase, clamp loader, single strand binding, all those things that we've learned about for replication to take place. But that's all triggered by the activity of SCDK, which basically reorganizes this complex, gets rid of CDT1 and CDC6, which were the helicase loaders, and then you get activation of your helicase, and you get recruitment of all your other proteins for replication. Who is raising their hand?
I was just had a question because in the last slide I thought you said phosphorylation by CDKs inhibits pre-RC formation, and now it's activated.
Yes, so SCDK. So remember, there's a different CDK for every phase of the cell cycle, and those different CDKs are going to have targets that are going to promote that phase of the cell cycle. So during S phase, SCDK is going to phosphorylate different things that are going to promote entry into S phase, and then in G2 or whatever, different things are going to be phosphorylated by that CDK. Okay? Other questions? I know it's a lot today. Are you guys doing okay? No. No. Okay. Okay, so then let's think of how we can, let's see. I think I have a discussion thing right here coming up. No, oops, wrong way. Okay. Oh, good. Okay. Yeah, I knew it was coming. So for the next 10 minutes, I want you to chat with your friends and discuss regulation of initiation of DNA replication in bacteria versus eukaryotic cells. And just go through what we just talked about. Spend 10 minutes thinking about it, discussing it, while I'm in the room with you, and so we can get questions answered. Okay, because I know it was a lot. Go.
That's
Yeah.
The thing is, like, if it's fully natural, it's like the opportunity to wander on
Yeah.
I mean, well, I mean,
you don't even
This stuff is always really down. Without making, I can do a whole entire class on which I'm actually There are conferences all over that conference.
I thought it was a tertiary. Or I thought it was a secondary. You're saying that was the
APC turns off, you know, I was doing that was too loud for a couple of years, but
Okay, did any questions come up? Do you guys want to go over this now, or do you want to reconvene on Thursday? Okay.